Hi everyone. So in our prior video, we talked about extravasation in the context of metastasis, where our kind of cancer cell is going to be exiting the blood vessel and entering this new environment. Our final step for metastasis is going to be formation of the secondary tumor. Step 8 is forming the secondary tumor. And there's kind of four main scenarios that happen when we form a secondary tumor. And we're going to walk through each of those scenarios. We're going to start with kind of the simplest one, which is going to be that when our cancer cell arrives, there's already a perfect environment. So let me draw my cancer cell. Let me try and write that again. There we go, much better. And the cell has just exited the blood vessel, right? So it just came from this blood vessel and it's entered this new environment. A perfect environment would look like lots of growth factors of different types that are present in the environment. It might look like immune cells already being present. Or some type of inflammation. Just generally, it's going to be a situation where cancer cells are going to be able to take advantage of the kind of existing environment to grow and thrive. So that would be our first example of an environment that a cancer cell might encounter when it exits the bloodstream. For our second example, if I kind of grab this and start to talk about this second example. In our second example, when we arrive as a cancer cell, there aren't immediately factors that are beneficial. However, at some point, there's a change in the environment. And it's not necessarily because we did anything, it's because of external factors. So some of those external factors that could improve the environment for our cancer cell would be inflammation, this could be due to illness or injury. This could also be carcinogen exposure. And it could be removing the primary tumor. Now, this last one might kind of feel a bit counterintuitive, but if we think about the process that the primary tumor has been going through with angiogenesis and metastasis, these processes are going to release a lot of inhibitory factors. This can include kind of that thrombospondin that's released that we talked about. There can also be just general inhibitory factors that we won't go into the details of that are released when we remove a primary tumor, or that are released when a primary tumor is undergoing angiogenesis and metastasis. During angio. Now, the primary tumor, because it's very established, is not really impacted by these inhibitory factors, but a secondary tumor that's just starting out is going to be heavily impacted by this. So then when we remove a primary tumor, these factors aren't really secreted anymore, and we're able to grow just a little bit more effectively. So any of those changes could lead to an improved environment where the cancer can now thrive. So our cell kind of just was dormant for a period of time, waited for these changes to take place, and when they did, the environment improved and it's able to grow and thrive. Our third scenario is a little bit more active on the part of the cancer cell. So this is where cancer improves the environment post arrival. So our cancer cell is going to arrive. I think I still have that copied. Perfect. Our cancer cell arrives. The environment's not great yet, but our cancer cell is going to do two things to improve the environment right off the bat. First, it's going to activate the stroma, so the surrounding cells. It's going to take control. And then secondly, it's going to produce growth factors. And it's going to tell the stroma to produce growth factors too. And in doing so, it creates a suitable environment after arriving. Now, kind of the most complicated and interesting situation is when cancer creates a pre-metastatic niche. Pre-metastatic niche. Awesome. Okay, so in this case, let me kind of draw out a diagram of what this might look like. So this is the primary tumor. Maybe it's located in the lungs. And then we have our secondary niche, maybe in the breast tissue. But we haven't actually sent any cells here yet. This is just a niche that's going to be a secondary site for us. So before our tumor cells even exit the primary tumor, our primary tumor is sending factors. And they kind of send a couple different things. They send tumor-derived secreted factors, which can include things like VEGF, which is promoting angiogenesis. It can also include TGF-beta. And it's also going to send exosomes, which are kind of going to be these bubbles of DNA and RNA and proteins. So these are our two things that we're sending out into the body. And these are going to go to our secondary site. And they're also going to go to the bone marrow. And the bone marrow is where most of our kind of immune cells are going to be created. So by sending these factors to the bone marrow, we're impacting kind of the immune cells that are going to be produced. Now, in our metastatic niche, our pre-metastatic niche, receiving these factors is going to lead to kind of the activation of the stroma. We increase the release of MMPs, and we're going to increase inflammation. So that's going to be the impact of these kind of initial factors that are being released by the cancer cell. Now, the bone marrow is going to send a couple different cell types over in response to the factors that cancer was sending out. It's going to send tumor-associated neutrophils, tumor-associated macrophages, Tregs, which are cells that regulate our immune system and kind of suppress it and keep it in control. And then we're going to get some myeloid-derived suppressor cells. And these are going to be sent over by the bone marrow and kind of in equal measure recruited by these active stroma to come to the secondary site. So it's kind of a two-way street where our bone marrow sends out these cells and then our active stroma recruits them to the site. Once these have reached the site, we're going to further activate our site in two ways. We're going to increase the secretion of growth factors. We're going to further increase MMP production. We're going to further increase inflammation. And we're going to engage in immunosuppression. So that way, when our cancer finally sends a set of cells over, this is our metastasis, it arrives and the niche is perfect and ready for it to thrive. So this is our pre-metastatic niche example, kind of our final example of how we're going to form a secondary tumor. So those are our four kind of ways of forming that secondary tumor. This was the final step in metastasis. Yay! So thank you for watching these videos. I hope you learned something. And yeah, let me know in the comments if you have any questions.